introduction. So while coming to the introduction, ovarian cancer is one of the leading cause of cancer death among the gynecologic malignancies worldwide. So here the rate of prognosis and recurrence is very poor even after the disease is treated. So in this case, polymers have shown excellent cytotoxicity, high functionality, and good protein resistance towards some applications like drug delivery, tissue engineering, and protein therapeutics. So among the strategies of drug delivery system, here peptide drug conjugates have shown considerable promise. So researchers have developed an expensive drug delivery system by using some characteristics, stimuli sensitive nature, thermosensitive nature, which is pH and temperature sensitive in nature. So here we have selected elastin-like polypeptide. It is a carrier and acts as a good carrier to various kinds of cancer therapy. So elastin-like polypeptides, they are biodegradable, non-immunogenic, and retained in blood circulation for a longer time. The most important feature in this peptide, they exhibit inverse temperature transition behavior, and this improve the drug pharmacokinetics and lead to increased tumor accumulation over free drugs due to passive targeting, which is called EPR effect, that is enhanced permeability and retention effect. So what is EPR here? It tends to accumulate only in the rather than the normal cells. So then coming to the next slide, we say like problems associated with the drug-mediated chemotherapy. The first main reason is when we deliver the drugs to the tumor, it remains a critical problem in the cancer treatment. And when achieving the desired therapeutic drug levels within a tumor is very difficult. Why? The main reasons are low molecular weight drugs have a tendency to get into all body parts and rapidly excreted. Achieving the desired therapeutic drug levels within a tumor is problematic, and there is a repeated doses and toxic side effects. These are the main reasons where the drug-mediated chemotherapy. So why peptide drug conjugates in cancer chemotherapy? To overcome the problems which is associated with the drug-mediated chemotherapy, researchers have investigated newer therapies. In that therapies, this drug delivery systems have been carried out, and this is mainly focused to target the drug only to the site of action without affecting the normal cells. So because of this point, this peptide drug conjugates have become an important area of investigation. So why peptide drug conjugates? It actually plays a major role in delivering the drugs. And the combination of more than one drug molecule in a short peptide drug conjugate is biodegradable through a linker. And the main essential parts for this peptide drug conjugates means to design it, we need a drug, a linker, and the peptide. So coming to the thermosensitive peptides, temperature-sensitive peptides, it exhibits lower critical solution temperature and inversion temperature behavior. This means where the temperature in the cancer environment, it gets more higher than a normal body temperature. So particularly at that temperature, the drug gets delivered. So that's why it is called temperature-sensitive peptide. And this is aqueous in solution when it is below 37, and when it is above 37, these peptides aggregate. And peptides are biodegradable. They also have it's a linker that is based on pH sensitive peptide here. So here the pH of the tumor sensitive, it gives some response to the changes in the surrounding medium, which is tumor environment, and it also expand or collapse depending on the pH of the environment. And why there is a change? It also changes by means of some function. I have selected elastin-like polypeptide. What does it mean? Elastin-like polypeptide is genetically designed as VPGXT. This is valine, proline, glycine, and X. We can select any amino acid except proline because it neutralizes the characteristics of the other amino acid. That too, especially it also can be N times. So it undergo inverse temperature transition behavior in response to a temperature change. And this inverse temperature transition behavior can be exploited for targeting purposes, as the temperature at tumor is slightly higher than the normal body temperature. So this is a drug, gemcitabine, and the molecular weight of the drug is 263.18, and the chemical formula. So this is the linker NH2. This linker is hydrogen, and which it plays a major role to release the drug at the specific target only at the specific condition which is acidic in the nature. The main feature of this hydrazine is this is the fact. So for the hypothesis, when we go for hypothesis, this is the elastin-like polypeptide, and in fourth place we have selected valine, and this peptide is conjugated with a drug here, with the anti-cancer drug, and by through EPN effect and the temperature simulation effect, the drug is loaded into the cancer cell, and here the peptide degrades and the drug is released in the cancer cells. So based on our hypothesis, we have aimed to synthesize a thermosensitive and pH sensitive elastin-like polypeptide and conjugate the drug to target tumors. And the main objective is to synthesize elastin-like polypeptide and conjugate the drug to the peptide and characterize the peptide and the peptide drug conjugate. And we have performed in vitro release parameters and also the molecular mechanistic pathway of the peptide drug conjugate through gene expression studies. So the plan of work was stage 1 is synthesize. We have to synthesize the elastin-like polypeptide. We have to conjugate the drug. And the stage 2, we have done the in vitro cytotoxicity study by using cell lines, ovarian cancer cell lines. And in the stage 3, we have done the gene expression study. So here we, as I told, in the fourth position, we have selected valine and the drug is gemcitabine over here. So this is a sequence which we have designed because it is shown in the color where V is blue, proline is green, and glycine is red. And again, valine comes blue and the glycine is red. So we have to synthesize the peptide here. For each and every group, we have to protect the amino group first for all amino acids, valine, proline, and glycine. So for the protection of amino acid here, we have selected the FMO chloride here. And when it is reacted with FMO chloride, we get the amino group protected valine. So this is the FTI spectrum where we have done the confirmation study whether it is protected or not, and this is the FTI spectrum of FMOC valine where we can observe the particular region in particular group in this region. So when we go for the second amino acid, proline is also reacted with the protecting, we get the amino protected proline. So we have got the confirmation study by FTIR, and these are the region where the groups are present. So when we go for the last amino acid is also reacted with the protecting group, we get the amino group protected glycine, and the confirmation is done by FTIR, and we can observe the groups which are presented in the structure. So apart from that, we have done the confirmation by TLC and in hydrogen test. So in hydrogen test, we have to see whether the valine and glycine we observe in deep blue and proline in yellow. So we have observed that, and the confirmation have done the synthesis of peptide for the first phase protection of amino group. So now after the protection of amino group, we have to use a resin which is supported with a protected amino group to go for the main synthesis. So here the 1,1 amino acid we have to protect and deprotect. So when first step is protection, so after the protection of first amino acid, we have to deprotect the agent. So we get the first free amino group here. So the first free amino group is again coupled with the second amino acid, and after this deprotection, we get the second free amino group. And the second free amino group is coupled with the third amino group, where after deprotection, we get here the third free amino group. The third free amino group is coupled with the next amino acid, and this is deprotected. We get the fourth free amino group, which is a secondary amine here. And this fourth is coupled with the last amino acid, and after deprotection, we get the fifth free amino group. This is the sequence which we have designed as I told earlier. So we have done the confirmation study for each coupling and deprotection where we have observed the results according to the inference. So we have confirmed the results first with the observation, and we have done the exact mass spectrometry for the peptide where we get for this synthesized peptide, the exact molecular weight is 427, and we have observed the peak in mass spectra whether this is a successful coupling. And here these are the rest. So here you can observe many peaks here. So these are the fragmentation for other peaks. And when coming for FTIR, we have observed all the regions here. In particular region, we have observed all the groups here. So this is NMR nuclear magnetic resonance H1 and C13. The same way, all the groups are present here and we can observe in the particular region here. So all these groups are observed, and in NMR we can see as a doublet, this is like a singlet peak, and when it comes here, this is a multiplet peak, and when it comes here, it's a doublet peak. So according to the region, we have observed the peak. So when it comes to C13 NMR, the same wise, how many carbons are present here, we have observed, and we have marked it as from C1 to C12, and we have got the carbon peaks. So to release the drug, the acidic pH, we have to confirm whether the peptide has got the pH below acidic. And we have found this in an online software IPC Python, where here we get the isoelectric point is 5.98, and it shows, it clearly shows it is below acidic in nature. So when we go for the transition temperature, as I told earlier, the transition temperature is the most important effect in peptide, and it should have the transition behavior from 37 to 42, and we have observed the peak here at 42 degrees. So coming for the phase 3, once all the synthesized peptide is synthesized, we have to conjugate it with a linker where it should release the drug at the acidic pH, and we have conjugated with the linker, we get this group, and the confirmation is done by FTIR, and we have observed all the regions in particular region, all the groups in particular regions, and here this is a molecular weight of the particular group. So we have observed the peak at 281, where peak molecular weight is 278. It can either be plus or minus 5. So at the last step, we are going to conjugate the drug with this peptide. We get this group, and the confirmation is done by mass spectra, and the whole group, the molecular weight is 686.72, where we have observed the peak at 686. And for all these peaks, we have done the mass fragmentation. We have observed the peaks. And coming to FTIR, all the groups are observed in the particular region. And in H1 NMR, we observe singlet, doublet, triplet, and multiplet for all hydrogen atoms. And when coming for C13 NMR, we observe all the carbons in particular regions from C1 to C20. We have observed all the carbons present here in nuclear magnetic resonance. So before going for in vitro cytotoxicity study, we have done the calibration curve for drug in acidic pH, which is 5.4, and basic pH is 7.4. So once if the calibration curve is done, we have gone through the drug release study. We have performed it through acidic pH and basic pH. So here in acidic pH, we have done the study, and we have seen 50% of drug was released and for 12 hours. And when coming to the basic pH, we have observed 11% of drug. So here we can say like our synthesized peptide is pH in nature, so that in acidic pH it has been synthesized. So when comes, this is a drug release profile study. So here you can see the pH 5.4 and pH 7.4. So when coming to the cell line study, we have used ovarian cancer cell line study, and we have calculated the IC50 value for the drug, peptide, and peptide drug separately. So here, when coming to the peptide drug conjugation, at lowest concentration 1 or 2 microgram per ml, it is able to inhibit the ovarian cancer cell lines. So from this graph, you can see like the drug peptide compared with the drug, where at the lowest concentration itself is able to inhibit the ovarian cancer cells. So this is most effective in killing cancerous cells. So when coming to the mechanism of action of the drug gemcitabine, here we can see this gemcitabine is converted into gemcitabine monophosphate, and this gemcitabine monophosphate is converted to gemcitabine diphosphate, and again it is converted to triphosphate, where we get CTP synthase 1. And here this gemcitabine diphosphate, it inhibits ribonucleotide reductase by reducing the conversion of CTP. So here we have observed in literature survey the overexpression of gene NT5C3 is overexpressed in this particular enzyme. So we have selected that particular gene for the gene expression study. So why it is NT5C3? It is mainly associated with this drug chemosensitivity. And it also provides some biological evidences in support of their involvement in variation of gemcitabine cytotoxicity. So when we see here how to perform the gene expression study, we have found the IC50 value in our in vitro cytotoxicity study. So we have to do primer synthesis, RNA isolation, cDNA synthesis, and RT-PCR by delta CT method. This is a method where we can get the results for this gene expression study, and these are the formulas which we are going to use. So we have done the primer synthesis. So before starting primer synthesis, we have used the controlled gene GABPDH, and the gene which we have selected is NT5C3, and we have to select the forward primer and the reverse primer for the gene expression study. And we have to do the RNA isolation, and we have calculated according to the formula, and microgram per ml we have got the results. And when coming to the cDNA synthesis, we have also done this in a mini kit, and we have got the results in microgram per ml according to the formula. So when coming to the real-time PCR, this is the main experiment for the gene expression study. So where here we have got the delta CT and the gene expression values. So here it clearly shows this peptide drug conjugates downregulates the NT5C3 gene, which is observed in the drug, than the peptide drug conjugate, which is very lower compared to the drug. So this is more effective in the gene expression studies also. So in coming to the summary, we have designed the synthesized sequence. We have observed, after synthesize, we have observed the sequence which is same, which we have designed. And after conjugating and after doing the characterization studies by FTIR, NMR, LCMS, we have observed the result exactly to our objectives and the aim. Where here in aim and objective, we have told the transition temperature of peptide, it should be 37 to 42, and we have observed exactly at 42. And we have mentioned it should also be a pH sensitive nature. So below basic, we have got acidic nature 5.982. And in vitro study, we have observed in acidic pH, it releases more % of drug than the basic pH. So it confirms the study. And when coming to the in vitro, found the IC50 value, and at the lowest value, it releases the drug. So it also confirms through cytotoxicity study. And when coming to the gene expression, we have observed the lower value compared to the drug where it is treated with the ovarian cancer cells, and the gene expression study is also confirmed here. So finally, this result prove it is also thermosensitive because we have got the transition temperature, and it is also pH sensitive, and it could deliver the drug at the particular site. So hence, this synthesized peptide drug conjugate will definitely offer a great advantage in drug delivery in response to stimuli in internal and external microenvironment. Thank you.